So the central dogma of molecular biology explains how genes are expressed, how we go from this genetic material in the DNA to the protein, which is the gene product. So sometimes we want genes to be turned totally on or totally off. Sometimes we need to have them on a dimmer switch where we want some, but not a lot. Sometimes we need a lot, but then we need to turn it down. Okay. Okay, so the dimmer switch is how we can turn the amount of proteins up or down so that we can figure out how much we need to have. We can have differences in cell differentiation. So your eye cells are going to express different genes at different amounts than your liver cells. Okay, so sometimes we need like a lobby versus the actual theater. Okay, but we need to have different amounts of light in those two situations. Differences in gene expression at different developmental stages. So this is kind of the same lobby versus theater things. And then it also allows us to change, to kind of acclimate to different changing environments. So for example, in my wilderness first aid class that I just had recently, we talked about how within 2 to 3 hours of being at a higher elevation, your body will start producing more red blood cells to carry that oxygen around your body more efficiently. Okay. And so we need to have changing. We need to say, oh, okay, we're at a higher elevation. We need to start making more hemoglobin. We need to start making more of these cells. We need to start that we can adapt and keep this organism alive. We're going to talk about the various levels of gene expression. Okay. This is not in your book, but it's important that we understand this because it helps us lay this out conceptually over that central dogma of biology. So again, this is why you're going to want to add these to your note card because it's going to help you. In eukaryotic organisms, there are 5 stages of gene regulation, of how genes are controlled and expressed at different rates. Okay. So first, this is the epigenetic control. So we're not changing the sequence of the A's, C's, G's, and T's in the DNA. We're just changing how available it is to go through the process of the central dogma. So we can have transcriptional control. So as we're going to control how much RNA is actually being produced as it's being produced. We can have post-transcriptional control. So how long is that RNA molecule going to last in the cytoplasm? Because the longer an mRNA molecule can last, the more gene product we're going to get from it. We can control gene expression as the protein is being produced. So we call that translational control. So during the actual building of the protein. Okay, so in epigenetic control, we're controlling the availability. In transcription control, we're determining how much mRNA is being produced. In post-transcriptional control, we've built our mRNA molecule. We're going to figure out how long will it last. Okay, translational control, this is as the protein is being built. And post-translational control, this is modifications that happen to the protein after it's been formed. The control over gene expression in prokaryotes is a little bit, there's fewer options because prokaryotes, the processes of transcription and translation occur at exactly the same time. So therefore, if we want to regulate how much protein is being produced, we have to also regulate what's going on at the transcription level. So, right, so if we want to control how much proteins are being made, we have to control how much mRNA is being made. Okay, so there's a whole bunch of environmental factors that go into this: the concentration of nutrients, the types of nutrients, how much oxygen is available, and you could go on, are going to determine how many enzymes are being produced and to what degree. Okay, so this is a big thing. The big takeaway for this slide is for prokaryotes, transcription and translation occur at the same time. Transcription and translation in eukaryotes are separated both time and space. Transcription is going to occur in the nucleus of the cell. This is where we're going to go from DNA to RNA. Okay, we have some time to modify our mRNA before it actually leaves the nucleus. Okay, so our process of translation is going to occur in the cytoplasm or wherever those ribosomes are present. Okay, so this might mean that there's ribosomes on the nuclear envelope. It might be ribosomes, ribosomes in black. Ribosomes that are on the rough ER or ribosomes that are just floating freely in the cytoplasm. Okay, so we need the ribosome in order for translation to occur.